Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing Central University's Common Entrance Test, that is CUCET entrance test for the MSc entrance, and this was for the subject Plant Biotechnology, conducted in the year 15th June 2023. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers being solved for this particular subject, you can go to my channel Omni Bioentrance, see for the playlist, select CUCET folder in the playlist. You will find the question papers answered there. Then next one. The conventional method to produce homozygous plants is lengthy and requires 7 to 8 recurrent cycles of inbreeding. Yes. The advantage of the homozygous plant obtained by diplodization of haploids include. Now after obtaining homozygous plants, you can convert them into diploids. So the advantages of the homozygous plant obtained by diplodization of haploids include. So homozygous plants which are can be converted to diploid. So what are the advantages? Detection of recessive mutants, of course. Production of stable homozygous dihaploids, yes. Detection of dominant mutants, no. Pollen culture, yes. So A, B, D only. Answer is option 2. Then next one. Essential step for protoplast development is removal of cell wall without damaging the cell constituent. The cell shows the following features. Now the protoplast, the cell wall itself is removed. So it will result in irresistible expansion of protoplast. First answer itself is correct. The isolated protoplasts are automatically sturdy or strong. No, that is not the correct answer. Large population of variable protoplast amounts are required. Nothing like that. Generalized guidelines for cell wall removal for all plant cells are applied. No. So the answer here is option 1. Then multi-step conversion of nitrogen to ammonia requires. So in nitrogen cycle you require 16 ATP. Answer is option. Then match the following. Growth regulators 0.001 to 10 micromolar amounts. Auxins. Auxins are elongation of stem tropism rooting. Cytokinins are BAP, benzyl aminopurine and zeatin. C2. Ethylene, it inhibits the growth but also promotes somatic embryogenesis. So the answer here is option 3. A3, B4, C2, D1. Then two statements are given. Shoot meristem culture was first applied to obtain virus-free plants. Yes. This we read just previously for last time for another of the question for the same entrance exam. Shoot meristem cultures are potential alternatives to more commonly used methods for serial regeneration as they are less genotype dependent and are more efficient. That is also correct. So both statement 1 and 2 are correct. Then next one. In somatic embryogenesis, presence of auxin lead to callus differentiation into proembryonic masses which develop into oliglobular stage. Yes, that is correct. Removal of auxin allow the differentiation of proembryonic masses further into globular, heart and torpedo stages to form dicotyledonous embryo. That is also correct. So both statements 1 and 2 are correct. Then next one. Trichoderma viridae could be used for biocontrol of the pathogen. So Trichoderma viridae, they had asked a question previously for Fusarium. But here they have asked for biocontrol of the pathogen. So it can be used not only for Fusarium but also for Pythium. So the answer here is option 3. Then next one. Hybrids are unique cytoplasmic hybrids that could be obtained using fusion of normal protoplast from one parent with enucleated protoplast from another. That will result in hybrid. A is correct. Enucleated protoplast can be obtained by radiation treatment. That is also correct. Then fusion of protoplast from one parent with protoplast containing non-viable nuclei. Yes, that will also result in hybrid. One of the nuclei should be proper, the other should be non-viable. That is how you have to obtain hybrids. Then D, if you see fusion of cells after subjecting both to gamma irradiation, both will result in non-viable nuclei. Then what will be the result of using both of them? No result. You won't get any hybrids. So D is not correct. E, fusion of one normal cell with one of gamma irradiated cell. E is correct. So A, B, C, E. Answer is option 1. EBAC. Then next one. Callus culture obtained from the infected viral plants cannot be raised as free from virus. A is not correct. Callus obtained from the infected viral plants cannot be raised as free from virus. That is not correct. The virus requires plant cells for replication. It is not able to divide, and as the cells continue to divide and increase in number, the titer of the virus comes down. The reduction is dependent on the rate of calli division and the time of subculture. That is correct. So answer here is option 4. That is A is not correct but R is correct. Then next one. Microscope. Microscope is immature pollen A3. Colchicine, it is a chromosome doubling agent B1. Answer is option 2. Then casein hydrolysate it is a source of a mixture of amino acids added to culture medium. Pollen is the male gametophyte. So answer is A3, B1, C4, D2. Then next one. Ploidy levels that are unstable include those where it is lower than that of the species. On the other hand, the ploidy of higher levels are usually more stable. Correct. The ploidy is related with the change in the genetic information inherited by the progeny. R is not correct. So A is correct but R is not correct. Answer is option 3. Then next one. The micropropagation provides the advantage where commercially many forest fruit trees, medicinal plants, ornamental plants are propagated. Of course, the distinct feature of these plants are, so micropropagation, it results in a clone of plants. So plants are clonal in nature, which are in large number and are called propagates. First one is the right answer. Plants obtained have unstable genotype. No, that is incorrect. Soma clonal variations are not seen. They are seen. They are prone to infection. No. Then next one. Quick wilt disease of black pepper could be treated by biocontrol agent. Yes. The two fungi genera Trichoderma and Gliocladium are found to be effective. Yes. Only glycadium is found to be effective. Yes. Quick wilt disease treatment by biocontrol agent is cheaper than the chemical method. Only trichoderma could be used for treatment. So A, B, C only are the correct answers. Answer is option 1. Then next one. The tissue culture medium commonly used for anther culture is the formulation of niche and niche. Answer is option 2. The most popular media formulation for culture of protoplast is B5. Answer is option 3. Crop improvement through tissue culture is possible if the explant regenerate into a complete plant. Only then crop improvement through tissue culture is possible. If it give rise to callus, it is of not much use. Then roots, you cannot take only the root and plant it and see that you will get the plant developed from there. No. Leaves, no. So it has to be a complete plant. So answer here is option D. That is option 4. Sorry, not option D, it is 4. D only. Which of the following factors do not affect the shoot bud formation? Do not affect. Culture medium composition. Yes, they will affect shoot bud formation. Genotype of the plant. Yes. Choice of the explant. Yes. Method of transformation of callus obtained from the explant. Yes, that will not affect the shoot bud formation. Float culture method is a well established technique for. So it is spontaneous dehiscing anthers by using L-serine. Answer is option 4. Then next one. Protoplasts are ideal system for manipulation of cytoplasmic genes and in the making of hybrids. To obtain protoplast, one need to remove the cell wall of plant cells. It can be achieved by treating the cells or tissues with cellulase pectinase mixture. This will remove the cell wall. Answer is option 3. Pathogen associated with molecular patterns PAMPs are. They are non-immunogenic response of plants. Answer is option 2. The axillary buds are pre-existing meristems. Yes. Have advantage of true to type progeny. Yes. Application of cytokine will stop the growth of axillary bud. No. That is not correct. Application of cytokine will not stop it will enhance the growth of axillary bud. Application of cytokine will enable axillary bud to grow in presence of apical buds. That is more correct. Axillary buds show variation among the different plants. Yes. So A, B, D only. Okay, that is seen in option 4 only. A, B, D. Auxins are involved in many processes associated with plant development. The synthetic auxin include indole 3-acetic acid. Answer is option 1. By benzaminopurin and creatine are cytokines. Diphenylurea, it is a urea-based plant growth regulator and not an auxin or a cytokine. It stimulates cell division and growth, enhances root development, promotes shoot elongation, increases fruit set and yield. Then, some vitamins are essential components of tissue culture media. They are essentially involved in. They have growth regulator functions. No. Dehydrate the medium. No. Hydrate the culture medium. No. Have catalytic function in enzymatic reaction. Yes. Answer is option 4. The method used for somatic cell hybridization. Somatic cell hybridization, it is a technique to combine the genetic material of two different species or varieties by fusing their somatic cells. So in this process there is cell isolation, then protoplast isolation, then you fuse them with polyethylene glycol, or at high pH and calcium. Then hybrid cell formation take place. Then culture and regeneration. Electroporation, no, it is not involved in somatic cell hybridization. High pH A2 positive, yes. PEG polyethylene glycol, yes. Particle bombardment, it is involved in plant transformation. Micro injection also involved in plant transformation. So B and C only. Answer is option 3.